It is certainly a delight to be here, and I thank you for including me in this prestigious meeting. I'm going to talk to you about the concept of gender and its relationship, as I see it in any case, to personalized medicine. The profound alterations in the physician's view of human physiology over the past 2 centuries of medical investigation have evolved in 3 sequential phases. Each is a reflection of and a direct result of societal attitudes, mores, and the cataclysmic events of human history. Pari passu, our sense of the rights and responsibilities of both the human subject of medical investigation and of the scientist pursuing a more precise understanding of personal physiology, have inevitably and fundamentally changed. Historically, for 25 centuries, we regarded humans, with the exception of their reproductive biology, as essentially interchangeable. We assumed that men and women had identical physiologies. The medical research of the latter half of the 20th century introduced the realization that men and women are not, in fact, interchangeable, but significantly different, establishing the relatively recent discipline of what we call gender-specific medicine. That is, the detailed study at the clinical level of the differences between men and women's normal function and their experience of the same diseases. The new approach produced a cornucopia of data, the extent and significance of which were completely unanticipated. Most recently, the stunning discovery of the structure of the human genome, over 2 decades in the making, created an entirely new paradigm for understanding human physiology. Pundits predicted that a detailed description of the individual genome would not only reveal the uniqueness of each person, but eventually, if not immediately, guarantee the optimal tailoring of medical treatment to each of us. That initial euphoria has been replaced by the realization that the task of delineating the molecular machinery that establishes and maintains the phenotype is huge and almost intractably complicated. The comments in the landmark 2001 paper from Venter's group that described the sequence of the human genome are worth reviewing. Essentially, the authors warned that this achievement was only the first level of understanding of the genome. They commented that the abundant features and complexity of human development could not be explained by a simple gene equals function analysis, but depend on what they described as a complex system of genetic regulation. They say, neither gene number, neuron number, nor number of cell types correlate in any meaningful manner with even simplistic measures of the structural or behavioral complexity of the individual. Nor would they be expected to. This is the realm of nonlinearity and epigenesis. Finally, they warn, there are 2 fallacies to be avoided: determinism, the idea that all characteristics of the person are hardwired by the genome, and reductionism, the view that with complete knowledge of the human genome sequence, it is only a matter of time before our understanding of gene functions and interactions will provide a complete causal description of human variability. The problem of translating the molecular biology of the genome to the phenome, however, is proving to be orders of magnitude more complex than what was originally supposed. For example, the genomes of different individuals differ by only 1 to 3%. Is this enough to account for the myriad of differences between individuals? Certainly, it does not account for the sexual dimorphism between males and females, much less the gender-specific features of the experience of the same diseases. Physiology, as it turns out, cannot be defined simply by the equating of gene structure to function. Over the last decade, however, the expanding science of how genes are regulated provides insights into how this is achieved, as Di Piero comments in a just-published essay. He says, "The eukaryotic genome is far more than a simple DNA polymer that encodes the sequence of proteins. The genome is, in fact, an information processing machine that helps regulate the transcription of the very genes encoded within the DNA." What then is personalized medicine? Broadly considered, genomic architecture is only one component of personalized medicine. In fact, it is an example of systems biology wherein genomes, transcriptomics, proteomics, and metabolics are combined with data about the individual's lifestyle, cultural, and social and environmental factors and beliefs, his disease profile, and a general phenotypic description. It is an encompassing, highly detailed, and completely individualized approach to the patient. Moreover, it is not a fixed phenomenon, but is changing constantly in response to the individual's developmental stage, hormonal status, and environment in sex-specific patterns. It is essentially driven by the same principles that have always been applied to the clinical care of the individual patient in the doctor's consulting room. Those principles are increasingly amplified, focused, and made more accurate as a result of our increasing experience in establishing the molecular biology of health and disease. The current push to authenticate the concept of personalized medicine envisions a universe in which each individual can be completely characterized at a molecular level, and the prevention, diagnosis, and treatment of disease meticulously tailored to the unique features of that individual's physiology. To that end, we have constructed and made universally accessible enormous collections of genetic information based on the conviction that we will be able to delineate the precise genetic scaffolding that creates specific diseases and that accurately predict responsiveness to therapeutic interventions. Semenoff and his colleagues reported that reasonably current sources document more than 500 million biospecimens containing genetic material stored in public and private databanks. What is needed, however, is corresponding phenotypic information and the computational tools necessary to link genomic data with phenomic characteristics. Altman warns, the genome sequencing projects were neither unmitigated successes nor failures, but rather the start of a newly enabled era in which determining the sequence of 4 DNA bases is easy, but understanding its role in biological systems is incredibly challenging. Venter comments, the generation of genomic data will have little value without corresponding phenotypic information about individuals' observable characteristics and computational tools for linking the two. He correctly points out that collecting, much less keeping confidential, adequately detailed data about each participant in the genomic databases will be as complicated and challenging as defining the molecular structure of the genome has been. Connecting genomic biology with the enormous variability of population groups seems a virtually insurmountable obstacle to deciphering the relevance of genetic architecture to the physiology of all races and the subsets of the human race. Humans populate vastly different geophysical, societal, and cultural environments. Not only do they differ in biological sex, but it contains a whole sequence of variation between male and female, and the environment further modifies genetic expression. Some aspects of different cultural values and perspectives on the human condition, particularly with regard to the respective societal roles of and the widely different value placed on men and women, are virtually impossible to quantify, but profoundly impact health and treatment modalities. For example, Jafari and Moazzam report one trenchant comment from a Pakistani student. Gender images are created by society, and we spend our lives living up to those images. Those of us working in gender-specific medicine are asked repeatedly why the sex of the patient matters if we are able to identify the 3.3 billion base pairs that make up the human genome. Put simply, will the advent of personalized medicine, with its dependence on genomic analysis of the patient, make the consideration of gender irrelevant? This is a picture of the gonadomorphic finch, that amazing bird, which is half male and half female, although the hormones that circulate through the body of the bird are identical, and they show the power of sex in delineating the phenome. It is now apparent that personalized medicine does not make biological sex irrelevant, but reflects and reinforces its essential role in shaping the phenotype. The Promethean, sex-specific effect of gender on gene expression extends to every cell, tissue, and organ of the body. In defining the moral and ethical responsibilities of the physician-scientist, this is a crucially important concept. It is important to note that interpreting genomic data, which are demonstrably sex-specific, fashioning gender-specific therapy to address those differences must eventually follow. It is equally important for those involved in changing genomic structure to understand that the consequences of their intervention might be significantly different depending on the sex of the subject. Reports of interventions rarely, if ever, comment on this consideration. As our exploration of genomic activity progresses, we continue to expand our information about how it is modified by environment and experience, age, developmental physiology, hormones, and perhaps most unexpectedly, by biological sex. It is now widely appreciated that the sex-biased expression of genes is ubiquitous. The evidence is incontrovertible that sex governs gene expression, not only by virtue of the activity of the X and Y chromosomes, but by the sex-specific features of many autosomes. In a trenchant discussion of the importance of sex in biological research, Virginia Miller writes, "One must wonder then, why is sex of experimental materials so often ignored in an era of genomics and personalized medicine? Investigators universally accept the fact that sexual dimorphism is significantly fashioned by gonadal hormones, but all too often consider them the only force in delineating the unique characteristic of the two sexes. In fact, the action of the sex chromosomes themselves is essential and important agent in establishing and shaping the phenotype." Ober pointed out, Autosomal genes, heretofore assumed to be the same in both sexes, are in fact often the agent of differences between males and females. He writes, although genes with sex-biased expression are enriched on the sex chromosome, thousands of sex-biased genes are also found on the autosomes. Although the DNA sequence, gene structure, and frequency of polymorphisms on the autosome do not differ between males and females, the regulatory genome is nevertheless sexually dimorphic. Yang's observation confirmed this over a decade ago. Her group reported that indeed, even at the molecular level, sex-specific genes are expressed in the body's tissues with small but significant differences in the proteins they produce. Here we report, she writes, surprisingly widespread sexually dimorphic gene expression. Thousands of genes showed dimorphism in muscle, fat, and liver, hundreds in the brain. Yang's group found two other important facts. Sexually specific genes were highly tissue-specific. Moreover, in addition to the X and Y chromosomes, Yang's group also affirmed the existence of tissue-specific sex autosomes. There are some striking success stories in genomic research. Its application to cancer has been one of the most valuable. Biological sex has an important impact on the clinical experience of malignancy. Lee and al. Investigated autosomal non-coding regions and reported sex-biased mutations in the whole genome of over 1,900 malignant tumors, and demonstrated functional consequences of genomic sex biases on the transcriptome and tumorigenesis. Thus, the sexual dimorphism in gene expression contributes to sex-specific susceptibility to disease. That susceptibility, moreover, is often tightly related to developmental stage and thus by inference to hormonal profiles that change significantly over the lifetime. Natri and colleagues lament the omission of obvious regulatory differences between men and women in the risk for and features of the same cancers. In spite of the overwhelming data that susceptibility and clinical features of the same cancers are sex-specific, these investigators comment on the indefensible lack of a consideration of sex differences in the development of cancer therapies. There is definitely a significant gap between what we are learning about molecular biology of the sex-specific pathophysiology of disease and the application of those data to the treatment of illness. A more effective focus on collaboration between clinicians and molecular biologists to fashion better therapeutic instruments for the sex-specific prevention and treatment of disease is urgently needed. In a just published landmark assessment of the impact of biological sex on the susceptibility to disease in the journal Nature, Brunac and his colleagues published the data from the entire hospitalized population of Denmark, almost 7 million admissions over a 21-year period, and half of these were women. Although both sexes shared many of the same diseases, the incidence, age of onset, longitudinal pattern, and frequency of associated illnesses were distinctly different between male and female patients. The authors emphasized that their study was essentially a documentation of sex differences which existed irrespective of mechanical, mechanistic, molecular causes, links to differences in the environment, or biases in the healthcare system. In a recent communication with me, Dr. Brunac, who is the leader of this group, reported that Denmark has just established a state-run national genome center that will host, generate, and oversee genomic data in a country-wide fashion. He will head the Research and Infrastructure Committee of the center, and he writes, "These data will be linkable to all the clinical data in the Nature paper. Such innovative and targeted correlations are clinically very important. Genomic research has presented us with a singularly important advance in our understanding of how the environment shapes the phenotype. Over the last decade, the traditional conceptual division between the terms biological sex and gender have largely disappeared. Historically, sex was considered the hardwired biological characteristic of the organism, while gender encompassed all the environmental experiences and influences that shaped human behavior. The first years of seminars at the Office of Research on Women's Health at the National Institutes of Health were marred with striking confrontations between molecular biologists and experts in the social and anthropologic disciplines. The latter repeatedly criticized the biologists' disregard for the role of environment, culture, and experience in fashioning human behavior. In fact, it is now evident that the environmental sea in which the individual lives out his or her life modifies molecular biology by impacting the genotype with powerful epigenetic modification that change its expression. In a very real sense, then, genomic behavior is plastic, changing throughout the lifespan in response to the individual's age and sex-specific physiology, but also to his or her unique environmental experiences. Some of these changes are not only permanent, but are passed on to at least 2 subsequent generations, often in a sex-specific manner. Thus, this new expanded concept of personalized medicine provides us with a final common path for what were considered 2 quite separate modalities. It presents a compelling explanation of how we are not only able to adapt and compensate for what happens to us in the exterior world, but makes apparent the mechanisms by which we are also the heirs of the experiences of previous generations. Scott Gilbert offers a brilliant new construct in what he calls ecological development biology, and says, "This is a science where organisms and environments possess agency, and the genome is both passive and active. This is not an approach against genetics or big data. Rather, it demands a broadening of the scientific portfolio so that other perspectives are also included in biological funding and as appropriate biological explanation. It argues that the physical organism is critically important, but that the environmental context plays a large role in gene expression." Springer and her colleagues echo this view. We are not arguing against any biologically based male-female differences. Rather, we are arguing that the vast majority of male-female health differences are due to the effects of the irreducibly entangled phenomenon of sex and gender, and therefore this entanglement must be theorized, modeled, and assumed until proven otherwise. Not only is our conceptual view of sex differences changing in spectacular ways, so is our ability to generate the science to support those perspectives. The 8-year-old Common Fund's Genotype Tissue Expression program is expanding our awareness of the significance and mechanisms regulating gene expression. It is a very important step to integrating the importance of sex in the regulation of gene activity. The consortium operates on the principle that although genome-wide association studies have identified thousands of loci for common diseases, the mechanisms of precisely how those loci are related to disease remains unknown. Essentially, the program proposes a biobank of samples from the major tissues of 1,000 deceased adults. The complexities of recruitment and appropriate informed consent of donors and their families are challenging. One of the most elegant examples of how GTX can yield sex-specific information with an unprecedented degree of sophistication is a recent paper from Tukyanen's group. the investigators established that incomplete inactivation of the X chromosome in female cells varies between individual cells and extends to population levels across a wide range of tissues. They report that at least 60 genes were sex-biased due to escape from X chromosome silencing and hypothesize that escape elements are contributing to sex-specific differences in health and disease. Remarkable and exponentially expanding new technologies to explore the molecular mechanisms that underlie the maintenance of health and the susceptibility to disease are reinforcing the importance of biological sex in shaping the unique features of males and females. It is worth reflecting on the biblical account in Genesis of how Adam and Eve originated. God shaped Adam from a handful of dust, his intricate physiology an active, direct, and original creation. Eve, in contrast, was fashioned out of one of his ribs, essentially a clone of her mate. It is interesting to think that we have spent the last 10 decades establishing, with greater and greater confidence, how differently the expression of their nearly identical genes is uniquely regulated, producing sex-specific versions of human physiology. We are in the midst of an astonishing series of discoveries about not only how extensive those differences are, but of how they are created and maintained in the extraordinary fluidity of genomic economy. Thank you.
Thank you very much, Professor Legato.